It is altogether the concept of bispecific antibody constructs, and there are different formats how to do that. The first one that was actually explored is the BiTE construct blinatumomab, a very small compound that was now approved in the States as well as in Europe for the treatment of relapsed refractory ALL. Now the next level new development is now with the new bispecific antibody constructs using a full-length antibody, and the target is CD20, which is obviously expressed on lymphoma cells, and then on the other hand connecting then with CD3 on the T cells, which would then be the effector cells.
Which then leads to the Regeneron. It's 1979 is the one that doesn't have the catchier name.
Right. So that is a single agent compound, and this is a phase 1 trial that was now presented here, an update. The first data was presented last year at ASH, and this is sort of an update now on the progress of the bispecific single agent activity. So we are still in the dose escalation phase, which is good news because it really shows that this drug so far has been very safe for the patients. No DLT so far been reported, and with the 3+3 design, more cohorts have now been treated within this trial.
And then how's that in terms of, you mentioned the adverse events, but combining the BiTE with an anti-PD-1?
Right, I think maybe just we should first focus on now the monotherapy. So here the safety profile is very favorable after some adjustments to the dosing scheme have been done by actually coming in with a low dose for the first week. The second week you do an intermediate dose, and then you reach the target dose on week 3. So with that schematic, the rate of cytokine release syndrome, the severity has dropped from potentially grade 3s down to grade 1 in the vast majority of patients, which is very easily to be treated. And the second very important point is that this drug doesn't show any neurotoxicity in these patients, which has been described previously for blinatumomab targeting CD19. So it has to be seen if this is in a bigger cohort, this can hold up that there's no neurotoxicity, but from a safety perspective, those are clearly much more favorable than previously reported with blinatumomab, which had 25% grade 3 and grade 4 neurotoxicities. So talking about efficacy, so we do see after reaching the target dose of 5 mg per for the patients, and now dose escalating up to 12 milligrams, the target dose, that in follicular lymphoma patients, more than 50% of the patients are showing responses, even complete responses as single agent. In the aggressive lymphomas, which is the second group that has been recruited here, we also are starting now to see partial responses in these patients, but it's obviously an open question as we are escalating, as these responses will maybe deepen as we now are escalating and perhaps defining then the MTD before we then can have a small expansion cohort to look at the safety profile of the MTD dose and of course the efficacy in these 2 big cohorts of patients of interest.
So then we can move on to the combination.
The combination, the rationale for that is that in vitro data in animal experiments, as well as in other clinical experiments, there is some upregulation of checkpoint ligands, checkpoint inhibitors. So the question is really here, if you add a checkpoint inhibitor, would you increase the efficacy of a bispecific in these patients? So Regeneron has developed their own bispecific, own anti-PD-1 agent, and the first question was, does this have the same efficacy in Hodgkin lymphoma patients, and what is the safety of this and also in NHL? So in Hodgkin's lymphoma, 60% have an objective response, so it basically mirrors what it also has been seen with other anti-PD-1 therapies, which are now being treated for Hodgkin's lymphoma. And there's some efficacy, definitely less as single agent with the anti-PD-1 in non-Hodgkin lymphoma, in FLs, as well as in diffuse large cells. So, but in terms of safety, we saw something very new. That was, if patients were treated with P13 kinase prior going onto the clinical trial, these patients were at risk of developing severe autoimmune diseases. So we have now amended the protocol that patients with P13 kinase pretreatment are excluded from the clinical trial. So after establishing sort of the safety profile, now we are taking on the combination, but we are now in the early stages of that by actually taking on the cleared safety cohorts of the phase 1 trial with the anti-CD3/CD28 antibody construct. So at a very low dose, at 2 mg at the moment. That was the entry. And in terms of safety, I think one major question was, are we going to see more severe cytokine release syndrome? Are we going to see more cytokine production? Are we going to see more neurotoxicity? And all these 3 questions can be denied. There's no more cytokine production. There's no difference, at least in these cohorts that have been tested, in terms of adverse events, treatment adverse events. So overall, the safety profile of applying this drug in combination with the already reported CD3/CD20 construct seems to be okay. And now we're moving into dose levels of the bispecific where we're seeing also clinical efficacy in the phase 1 trial. And there's now to be seen, once we combine these both, if this also leads into perhaps a very favorable safety profile, no difference to the single agent, but potentially better clinical control for the patients of their disease.
Very encouraging numbers to build on.
Yes, I think so. Obviously, these early days, it's just a phase 1 trial, but I think the plans are, as we are seeing now, clinical efficacy, to go ahead and explore this maybe in more advanced clinical trials.